Hey Mr. Taylor, I hope all is well. You said we can do a video for a test, so here I am doing one. So yeah, this is the first part of question 2. So here we are talking about, this is the label diagram of the chemical structure of an amino acid. So this amino acid, as you can see here, is valine, and yeah. So what I'm going to talk about right now is how every amino acid has the exact same group here. So they have a carboxyl group, an amino group, and a center carbon and hydrogen. So since everyone's the same, when they do connect and combine to create a polypeptide bond and chain, which I will show you a little bit later, the hydrogen and the amino group connect to create a water, and then the water connects to create the polypeptide bond. So what this does is that connects them, and then the way that we get 3D shapes is from the bottom part and it being nonpolar, and other sorts of groups such as polar, etc. So what these do here is they create folds and connections, because aminos, they want to fold and create like hydrophobicity, for example. They want to negatives and positives like stick together. They want to have a structure that they're happy with in a way. So what that does is it creates your tertiary, secondary, and quaternary structures, and it creates the proteins that we know. So part 2 of the second question. So here, what happens in how tertiary, quaternary, and other structures happen is because of folds and stuff in the protein. So the reason why folds and stuff happen is because, like I was talking about before, there are principles in these aminos that they want to fulfill. So for example, this threonine here is hydrophilic, and the alanine is hydrophobic. So what might happen in this protein is the hydrophobic amino might move towards the center, and the hydrophilic might move towards the outside. Well, not might, it will, because that's how the molecules work. So same thing happens with all the other things, and this creates different structures and things like that. So essentially, the tertiary structure, as we know, it is, here, I'll pull it up here on the website, it is a mess, almost like just tangled headphones or something, and in that is a bunch of different structures. So as you can see, alpha helix and B-plated sheet is in here. So that looks like that. Moving here to tertiary structures are created by nonpolar things because they're hydrophobic, and here they're also created by hydrophilic because there's subends and stuff from the structures. Now some R groups, as it says here, form hydrogen bonds. So what this does is it creates folds and shapes in the protein, and that's how shapes are created, like 3D shapes. So it's actually really cool. So then here, yeah, it says bridge, and then that's essentially how that happens. Structure, we already know what that is. So the forces of attraction that create this are hydrogen bonding and hydrogen bonding that create the alpha helix and beta sheet. So these create the looks of it, and then those can be found in structures within actual proteins. Alright, so final part here explaining how the shape is important to the protein's function. So, like I was saying before, how certain areas connect with different areas. So we have this color-coded, as we know, because polar is blue, like the glutamine, non-polar is yellow, like the alanine, aspartic acid is the negative charge, and then the last one is positive charge. So as we know, positives and negatives attract, that might connect, the R groups will connect, and it will make for a different shape of protein. So what this does is it changes the function, as basically what this allows it to do is it changes the way it interacts with different molecules and structures inside organisms. So that basically changes the whole function of it. So an example of this is collagen. So collagen is shaped how it is, and what it does is it actually is a form of like the ligaments and stuff in your muscles. That's the best way I can explain it. So it's kind of like the ligaments and tendons and etc. in your muscles, and it's really cool. It's in bones, and it's so basically what it is a very strong protein that will be broken. That's why in areas and shapes and stuff, it controls what how strong of a protein it is, and collagen is ended up being a very strong proteins that we need for like muscles in our joints, and very strong muscles that we do not want to break. So yeah, that's the final thing, and thanks for listening.